Hello, and welcome everyone. In this podcast, we're going to begin our discussion of population genetics. Remember, up to this point, for the most part, everything we've talked about is the individual. How do genes affect this individual? Or we may have talked about this individual's specific cells, and maybe different kinds of cells. And we talked about how these cells divide. We talked about how this individual makes gametes, and then how that passes on the genes to the next generation. And we talked about his genotype or her genotype here for particular alleles, maybe it's big A, little a. And we set up these fun little Punnett squares. At least I found them fun. And deep down you probably did too. And we were able to make a prediction about his progeny based upon who he may have had children with. And this all makes sense, again, for the individual, how his or her genome affects themselves and their subsequent generations. But now what we want to do is we want to ask the question of what happens if you have a whole bunch of people in this population. All look a little different, but they're all the same species. And so this is what a population is. It's a group of individuals of the same species that live in the same area. So all of these people here live in the same area. They're the same species. They look a little different, but they're the same species. So that's what a population is. So we're going to leave the individual behind for now, and we're going to talk about this population. And the next thing I want to define is gene pool. That would be all of the alleles of every gene in a population. So all the genes that make up these different individuals, all the different alleles they have, that is the gene pool. And the way I drew this, you see many different shapes and sizes and colors, and so it's a very diverse. Okay, as we leave the individual behind and start thinking about populations, I also want to leave the human behind as an example. We'll come back to humans later, but let's think about a population of, say, something like a ladybug. And I'm going to draw just several ladybugs here. And within these ladybugs, they may look similar to you or I, but let's say that they have different patterns here. Maybe this has spots on it. Maybe this has more, looks like bars on it. Maybe this one's shaded in a lot. Maybe this one has a wavy line. And you're going to have some repeats too, but maybe you'll have some with more spots and some with less spots. Maybe you'll have some that kind of crisscross. So we see many different kinds of ladybugs in any given population. And we call this a polymorphism. Essentially, this means many forms. These are, well, I'll write many forms down. And these you can also think of as inherited traits. That display many varieties. That can be visually seen. So as we look at this and we take a closer look, we can see there are some differences. So why do we see these different polymorphisms in this population? One might predict one of these polymorphic traits might have some sort of advantage and might begin to predominate in the population. But in this case, they don't. We just have several trait patterns here that seem to coexist. But what causes this difference within this population? Well, the term polymorphic explains it. This explains why we see polymorphisms. Not surprising. When we see this term polymorphic, what we're really saying is that there is genetic variation in the population. Now, we just spent the whole semester talking about mutations and the effects of those mutations and other aspects. So this is not shocking news to you, I'm sure, that the reason we see different patterns is because there are certain genes that exist in different alleles. What's interesting is there appears to be no competitive advantage to these because we do see in roughly equal numbers of different kinds of varieties here. When we talk about genetic variations, what we're saying is that there's greater than one allele per gene within this population. Now, for a comparison purpose, let's talk about a monomorphic. We get this when there is greater than 90% of the population has the same allele. So you might think in a situation like this, what you would have are ladybugs, and we're going to stick with our ladybug example here. They would all look the same. And that might be true. They might all look the same because they have the same alleles. So maybe I'm just going to give them one spot here. And because it's mainly this one allele, we don't see much variation here at all. Now it's possible you could still see some variation, because remember, the environment and other genes may contribute to some differences here. So maybe we do see every now and then something that looks a little different. But we'd still call it monomorphic because more than 90% of the genes in this population are of the same allele. Let's focus on polymorphic right now, because this is the normal situation. Alright, so I erased all that down there, and let's talk about how this population is polymorphic and how this genetic variation can occur. Well, I'm sure you can think of some ways, right? Because we've talked about mutations before. But you could have a wild type gene here that gives you a particular pattern of spotting on the back of the ladybug. You may end up with a deletion, or part of it's deleted. That could explain the polymorphic nature of this population. As you know, you could also get a translocation, something's added to it, or any of the variety of chromosomal changes that we've talked about. You could also get, these will all add to the genetic diversity within the population. It will deepen the gene pool, so to speak. However, the most predominant polymorphic change within a population are these things called single nucleotide polymorphisms. We commonly refer to these simply as SNPs, S-N-P-S. As I said, these are the most common. So let's write that down here, most common. They account for more than 90% of human genetic diversity. Diversity here, but in your notes write human genetic diversity. And all these are, and I'll draw it up here on our wild type strain, are single nucleotide changes. And we see them maybe one or two in this gene. We may see them outside of the gene, a coding gene. We see them in all different places. But they're typically just single nucleotide changes. Sometimes they have an effect on the phenotype, sometimes they don't. But they are changes within a population that adds to the genetic diversity. Now what if you wanted to calculate or quantitate this diversity? How would we go about doing that? So when we want to quantify this diversity, this genetic diversity within a population, a couple things we will want to do. We want to first look at or calculate the allele frequency. And how do we do that? Well, the allele frequency is equal to the number of copies of a particular allele divided by total number of alleles for that gene in the population. And we're going to work through an example of this in just a moment. We are also going to want to be able to calculate our genotype frequency. Okay, to calculate genotype frequency, what you do is you take the number of individuals with a particular genotype and you divide that by total number of individuals in the population. Let's calculate allele frequency first, and then we'll come back to genotype frequency. Okay, so let's calculate an allele frequency. In order to do this, we need one of a few pieces of information. You either need to know the genotype. Maybe the problem will specifically say these are our genotypes. That's not as much fun though. You may be able to determine genotype by looking at the phenotype. Third, you may have DNA sequence. You may have sequenced these pieces of DNA, and you know that. You know what the variation is because you know what the sequence is. That's probably the most direct way of doing this. You may also look at proteins, kind of like we did in class a while back when we looked at protein gels and we could see how far they migrated. We might be able to calculate genome frequency that way as well. So look at protein to determine. Alright, I want to look at this method here. Determine genotype by looking at a phenotype. So let's draw a population of ladybugs. We'll stick with ladybugs. And they're living at the base of this plant here, say. Living the dream that ladybugs often do. Okay, we have 20 ladybugs here. And let's say some of these ladybugs are red. And then let's say Some of them are pink. And the pink sometimes looks a lot like red, so for pink, I'm just going to put a P here. It is a pink color, but on the camera sometimes it doesn't pick up pink. So let's make pink like that. And then the rest are going to remain white, without any pigment. And so you might recognize this as being a case of incomplete dominance. Yes, we don't forget these things, right? They keep coming back to haunt us. And let's say we know that big R gives us a red color. And we know little r gives us a white color. And because it's incompletely dominant, a big R, little r gives us a pink color. And now, knowing this information, we can calculate our allele frequencies. So first, let's count the number of individuals who are red. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10 individuals that are red. How many white? 1, 2, 3, 4. We have 4 white ladybugs. And in pink, we have 1, 2, 3, 4, 5, 6 pink ladybugs. And so to calculate the frequency of big R, what we do is add up all the R alleles, big R alleles, and divide it by the total number of alleles. So what are the total number of R alleles? Well, in the pink individual, we know we have 1 R allele for every individual. So we know we have 6. We have a total of 12 alleles, but only 6 of them are big R. There are no big R alleles in the white ladybug. What about the red ladybug? Well, we know we have 2 big R alleles per red ladybug. So for here, we're going to go plus 10 plus 10, because we have to add both of these. And then we're going to divide that by the total number. And you can see we have 20 individuals. So we have 20 individuals. Each individual has 2 alleles. So we're going to have 40 total alleles. And this is going to turn out to be 26 out of 40. And we could break this down 13 out of 20, or put it in a decimal format or a fraction format. Either would be acceptable. I would prefer either a decimal or a percent. So 26 divided by 40 is equal to 65%. I'm going to write that up here. The frequency of R is equal to 65%. The frequency of little r should be 35%, but I think it's always good to go ahead and go through this whole process again, just to make sure we didn't make a silly math error. So the frequency of little r, big R red ladybugs don't give us any little r's. The white ones give us 2. There are 4 individuals, so it's going to be 4 plus 4. And then we get 1 per heterozygote, so we have to add 6. And what we get here, and we still divide by 40, and we end up with 14 divided by 40. And that does hold true and give us 35%. So the frequency of little r is equal to 35%. Now let's calculate our genotype frequencies. Now let's calculate the genotype frequencies. I think these are a little easier because we have all the data we need right here. So we know the frequency of big R is equal to 10, because we have 10 of those individuals, and we divide it by a total number of individuals, 20. So we know that's 50%. The frequency of little r is going to be 4 divided by 20, 4 white ladybugs divided by a total number of 20, and that gives us 20%. Then the frequency of the heterozygote, big R, little r, is going to be 6 divided by 20. And that's going to be equal to 30%. So this is one way that we could solve the allele frequency and then the genotype frequency. In class, we'll talk about another way we can do it by looking at the proteins using a protein gel. And then we'll also look at an example where we look at 3 different alleles. How do you calculate the allele frequency when there are 3 alleles in a population? Remember, there are usually more than 2 alleles for any given gene within a population. We often think of 2 and work with 2. Okay, I think this is a good place to stop this podcast. We just got done talking about allele and genotype frequencies. We also covered some general terms that are important for population genetics. In the next podcast, what we're going to do is we're going to take these allele frequencies and the genotype frequencies and try to decide if we can make any predictions about the population in the next generation. And we're going to introduce two new friends, Hardy and Weinberg. Okay, well, I'll see you in the next podcast. Bye.